Welcome back, everyone, to Translating Proteomics. Andreas and I have discussed AI and proteomics on the podcast before, and we've even had Matt McElwain and Vijay Pandey on to discuss how AI is changing the world of biotech. Today, though, I'm excited to have an in-depth conversation with someone who is actively developing practical statistics, machine learning, and AI tools for use in proteomics and systems biology, Professor Olga Vitek. Olga is the Raymond Bradford Bradstreet Professor and Director of the Barnett Institute for Chemical and Biological Analysis at Northeastern University. She's also the incoming president of US HUPO. Olga was previously a university faculty scholar at Purdue University, where she earned her Ph.D. in statistics. And I've been friends with Olga since our postdoc work together in Rudy Aebersold's lab at the Institute for Systems Biology. And I am so thrilled to have the opportunity to talk all things statistics, machine learning, proteomics, and more with her. Olga, welcome to Translating Proteomics.
Thank you so much for the invitation. I am thrilled to be here. I followed the podcast for quite some time, and I'm really excited to be part of the conversation.
Awesome. Well, let's start back in time. Let's start at the beginning. You're a statistician, and you're thinking I can apply statistics to anything. And how did you first end up in this intersection between statistics and biology in general, and then proteomics in particular?
This is like many things in life. A lot of this is just due to random chance and luck, but I was very lucky. So my first exposure to proteomics was when as a PhD student in statistics, I was a summer intern at Eli Lilly and Company. And I just landed by random chance in a mass spectrometry-based proteomics lab. And I was the only statistician in the lab. We were in the same office space with a chromatography expert and chemistry expert and biology expert and a database expert. And all of these people had questions to me, and I was just a student, and I was an intern, and I was hoping somehow to bring my expertise that I had at that time to help answer these questions. And so this is what really excited me from the first day on, because I felt like what I was doing was very useful and very necessary. And I really was happy to see that the results of our work had a very direct impact and very direct utility. And as part of this internship, my supervisor gave me some papers to read by a certain Rudy Eversold, with the first author of certain Alexei Nestrinsky.
Who
And
you
I
ended
felt
up,
like,
sitting next to you for several.
exactly, we ended up being office mates. And so when I was reading these papers, I was so excited because I felt like, whoa, these are life scientists and experimental scientists who understand that they need statistics. And maybe some of these methods can be improved a little bit, and maybe I can do a little bit better. But I certainly saw the utility of that.
Yeah.
And I just emailed Rudy and asking, can I be part of your lab? And, the rest is history.
That's that's amazing. And that was such a special moment with the introduction of peptide profit and protein profit. And I remember at that time in the field, there was a debate, there was an argument about whether or not we should be hand assigning confidences for identifications, or if instead we could use statistical tools to do this in a more automated manner. And it wasn't a foregone conclusion that
Mhm.
statistics were the right way to go.
That's right. That's right.
And so I'm curious, in that very first experience that you had at Eli Lilly, was it clear to folks immediately that statistics was a thing that was absolutely necessary, or was there resistance or uncertainty?
It was quite clear in a very fortunate way, because I think a really big part of statistical practice is statistical experimental design.
Yes.
And so when you design experiments in a way that accounts, for the properties of the data and for the uncertainty and variation and also for the systematic variation that exists, well, you end up with experiments that are much more informative and
Yeah.
they're much more resource conscious. And so when you come in and tell people, hey, I can help save you money, people tend to listen.
It's
And so I was
a good
very
selling point.
fortunate that my very first exposure to this was through the lens of experimental design. And back then, with my colleague Leah Reiter, we thought about how we can design experiments to and optimize mass spectrometry settings to improve, the number of IDs, identified peptides and identified proteins. And Leigh would be tweaking literally like the mass spectrometry. She comes from a lab that designed the instruments. And I was like, oh my God, I cannot touch that. But I
I
can
don't want
tell
to
you
break, I don't want to break the very expensive piece of equipment.
exactly, but I can tell you what would be the settings that would actually give us some information.
Yeah, that's amazing. And so tell me a little bit of let's talk about statistical experimental design a little bit because this is a thing that I feel like everybody should know about but doesn't. And so maybe you can give us the 1 minute primer
The reason why.
on yes. Yes, exactly.
Well, it's actually the reason why people don't really think about it that much is because it's actually not easy in the sense that you really need to have a good sense of how the data will be analyzed in order to design the experiment appropriately. So it is really not possible to design an experiment when you don't have a sense of the analysis. And increasingly our analyses are complex, right? The
Yes.
complexity of the data now matches the complexity of the problem we study. And so fundamentally, I think there is a gap between, continues to be a gap between people who do the experiments and people who kind of think about the analysis and machine learning. And this gap manifests itself very early on at the experimental design stage.
Mhm.
And I think that it is really not possible to ask people to be experts in everything because, the experimental part is complicated, the data analysis part is complicated. And I think a solution to that is really improving the communication between people who are experts in different fields.
Yes. So that interdisciplinary connection, because there is an entire field of statistical experimental design,
Of
which
course,
has a rich history, goes back decades,
the
maybe.
Fisher, yeah.
Yeah. Yeah. And and bounds, that people can bring in and I concepts of, oh, yes, this experiment is actually probably not going to lead to something in which we will be able to draw strong conclusions, which is a horrifying statement to have to say to somebody. Like, you could do this giant experiment that could take you years, and if it wasn't set up aligned with the way that it is going to be analyzed, it might actually be very hard to gain insight from that experiment.
That's right. And part of the communication gap also kind of goes both ways, right? So experimentalists need to kind of understand what is the mindset of experimental design, but also statisticians need to understand what the data are,
Yes.
right? Because it is really important to understand what is actually happening, what is controllable, what is not controllable, how different, like potentially minor, what looks like a minor detail in, sample preparation or separation or something else changes the nature of the data. And so it is really important to know also like what's possible and what the data look like. So really this communication has to go both ways. In my practice, I often find myself in a position of managing expectations, because there is no use saying, oh, you need like, hundreds of samples for something, right? So that
Right. You
because
need hundreds of
people,
samples, you need 2,000 replicates, and the
exactly, right?
experiment will cost you $400 million. And
Exactly. So that's not very helpful, right? But so what
yes,
we can do, we can kind of, yeah, manage expectations, saying, okay, well, given the resources you have and the type of variation you have, right? So this is what you can reasonably expect from this experiment. And then the question is it useful or not, right? And if it is useful, then by all means, right? But if not, then maybe we should change how we think about the problem.
Hmm. So this dialogue is really important and very I think the this question of how do we find an intermediate point where we are able to be confident about the statements we're able to make from the data, but that in the context of a designed experiment, that makes a lot of sense. Like I'm seeking to answer this question. I am seeking to compare A and B.
Mhm.
In the context of experiments where I'm gathering data from all over the world and
Mhm.
doing integrative meta-analysis of that, how should we think about uh, how experimental design and how statistical methods deal with that sort of situation?
Well, this is really challenging, but also very exciting because increasingly we start having solutions in this direction. And essentially to make some sense from what is called observational data. So essentially data that are not controlled by experimental settings, but data that come along either, like in a clinical context that samples just arrive the way they arrive, or maybe samples that are collected across different investigations in different places.
Mm,
We need to understand what are the sources of variation which can be, confounding the question we're asking.
yes.
And so I think it is really important to track the provenance of the measurements in the case of observational data, but also try to understand what are the sources, so both like from the nature of the samples and where the samples come from, but also from the nature of kind of biomolecular variation, what can affect the abundances of our readouts, right? Or something like that. And so if we can understand the sources of variation, even if we cannot control them, we
Mm,
can account for them. And so the first step is to really be cognizant of what can confound, what can affect uh, the measurements that we have.
mm,
And one way to do this in the kind of modern statistical machine learning methods is to work essentially with causal diagrams,
mhm.
which would try to make explicit our assumptions about the nature of the variation. And then if we have these causal diagrams, we can work with them in various ways. There are multiple flavors of statistical methods, which will either account for this confounding or will be able to tell you this experiment will only give you biased estimates of things you're interested in.
Yeah. The way I often talk about this with my students is, all right, you see an effect and you want to be excited about it. But on the other hand, there are 1,000 stupid explanations for
Exactly.
that effect. Like, what are all of those explanations? Because
Exactly.
and how can we be certain that it's not because, oh, this tube sat out for an extra hour
Hmm.
or um, this there was actually a case in our building where it turned out that there was a bit of a vibration in the floor and cells grown in one place versus another place experience that vibration differently. And this is not a factor that anyone particularly cared about, but it turned out that the cells actually noticed themselves being vibrated, and that led to an effect that was a systematic bias in the data, co-correlated with our primary variable, and really hard to untangle.
Hmm. That's right. And so I think that again, it ties to, okay, now how do we untangle that, right? So what is the analysis, right? And so what would be the design that would facilitate that analysis?
Mhm.
And so to me, the solution really is kind of going back to this communication gap
Mhm.
is interdisciplinary education
Yeah.
and making sure that experimental scientists have exposure to statistical methods and experimental design in a way that is accessible and non-threatening.
Mhm.
And at the same time that, computational scientists, they really understand the nature of the problems, right? And the nature of the challenges that people who do the experiments they have to face.
Mhm.
And
And
that
so
has been a big passion of mine to
yeah,
make
well,
sure.
I'd love to hear,
Yeah,
your top when people think statistics, their brain goes to t-tests and power analysis.
right.
But if you had if you could list your, top five things that you wish all experimentalists were aware of from statistical canon, what are those things?
Well, the first one is replication, randomization, and blocking. These are the three principles of statistical design. Well, okay, even before that, the very first one, talk to a statistician before collecting the data.
Oh, yes, number one. Yes. Uh,
Because everything else is already getting fairly technical, right? So knowing when to ask for help, I think this is probably the very first thing, right? Then the second thing,
every bioinformaticist I talk to is often in this position where someone comes to them with a collected data set and
of
is
course,
like, please find me, something interesting in my data set after I've collected it.
exactly, exactly. So yeah, the first thing is talk to a person who has a sense of how the data will be analyzed, right? And decide on experimental design in that way.
Mhm.
The second thing is, of course, the experimental design, because if the experimental is designed well, then we can kind of think about different flavors of analysis, right? And so replication, randomization, and blocking.
Mhm.
And then in terms of the data analysis itself, something that really concerns me increasingly is that, especially now that we have this new, very powerful methods, and neural networks, so somehow some people think that you have one class of methods that is good for every problem.
Mhm.
And it is not the case. So different questions require different methods.
Yes.
And so being able to match the question to the method, that's the next skill that doesn't have to be very technical, but needs at least enough intuition to understand that not every method answers every question. In particular, not every question is a t-test question.
Yes.
And not every question is a neural network question,
Mhm.
right? And so knowing how to understand how to match the method to the question, I think that's the next skill.
Mhm.
And then, the technical details, is it a t-test or ANOVA or, or MS stats, or something else, right? Or is it a neural net or logistic regression or neural network or something else? These things actually increasingly will be easy
Mhm.
because of all the resources we now have with AI, right? And all the implementation we have and all the, cloud code, etc., where you just kind of talk to a computer, then analysis happens. I think that this part is easy. And I think increasingly in terms of the education, we need to emphasize not just the technicalities of how do you calculate the sample size, but understanding what these methods do.
Mhm.
And most importantly, how these methods fail silently. So not fail in the sense of giving an error message, but
Yes.
fail in the sense of giving numbers that look very plausible and
Yes.
exciting, and you have very small p-values, and you have a small sample size, not realizing that in fact there is a like logical inconsistency behind the analysis. And so to me, the biggest kind of direction of closing this gap and the biggest kind of direction of interdisciplinary education is really in the understanding the concepts and the principles behind the methods more so than okay, here is like the package that you need to run with this,
Yes.
parameters.
Yes. Yeah, so the intuition and the why. Clearly, there is this huge AI explosion happening around us,
That's right.
and it is very easy to press one button and say, "Oh, I did a random forest, I did a CNN, I did an LSTM, I did a, like literally every alphabet soup of
Exactly.
statistical method. I ran all of those on your data, and here's what popped out."
Mhm.
And so I'm curious what your thoughts are on how we take advantage of the incredibly powerful foundation models, et cetera, while balancing that rigor and interpretability.
It is really hard. I don't have an answer to that in the sense that a lot of this is very tempting, what used to take months now can be done in hours.
Mhm.
And especially if the answers that are given look nice,
Mhm.
right? Yeah, you have some small p-values. You have, say, your favorite protein came somehow really important, right? And by the way, many of these tools, they're designed to please, right? So they're designed to actually give you the answers you are looking for. And so
Like, oh,
this
you've
all
studied
looks
P53 your whole life? Guess what? P53 is the most significant protein in your data set.
exactly, exactly. So this all sounds really good. And so how can we take a step back and look at this critically?
Hmm.
And so I think the answer really is kind of going back to what I said earlier. It's twofold. Once being able to translate the subject matter question, the biological question, or the biology question, or chemistry question, or clinical question, into kind of more the statistical question. So, okay, maybe we're talking about some populations, and maybe these populations have, mean abundances of this protein, and so what is of interest to us in terms of, their quantitative characteristics.
Hmm.
And then the second part would be to say, okay, well, what are the methods that are designed to answer this type of questions?
Yes.
And so if I'm offered, a random forest when my question is about clustering, so maybe I can question the nature of the answers that come through. So I really don't have a better answer than being able, like the education at the level that allows us to understand what these methods do
Hmm.
without necessarily going through, okay, here's a library, here's the parameter, here is the tuning something, right? But understanding at least the class of methods and how they match the questions. I
Hmm.
think this is really important. And then, well, the second, I guess, the third part of the answer, it is a little bit on us, right? On people like me and like my colleagues who develop computational methods. And this is something we think a lot about these days in the lab as we develop the tools. So currently people start interacting with these tools in a different way. So before it was, a PhD student or like a lab scientist kind of downloading a package, reading the vignette, right? Kind of figuring out, the error messages. Now there will be, a large language model saying, okay, I want to run this analysis. And so part of the thing that we can do is to design our tools in a way that interact correctly with this type of queries,
Oh, that's
that
interesting.
expose the right workflows and expose the right branching points
Hmm.
and help match the question to the method in a natural language manner. And I
Hmm.
think it will require us design our tools uh, differently,
Hmm.
but I think it is possible. And so
Yes.
that some kind of more or less standard or commonly used pipelines, they can be done correctly.
Yeah.
And the analogy kind of I think of sometimes is, cars. So when I was learning how to drive a very long time ago, I had to go through the school, which driving school, which had like a really substantial chapter on, car design, like how does the engine work and how does, the
Your
transmission
your
work.
your driving experience was entirely different than mine. I
Well, it was probably in a different context. But anyways, I had to take a test on the car design because, well, you may be stuck, in the middle of nowhere and have to change your own tire, and whatever, right? So they were preparing us for that.
yes.
Obviously, I've never used this type of knowledge and I completely forgot what it is. In part because cars are so sophisticated now, there is no way to have like a few hour class to tell you how it works. But
Yeah.
also because the infrastructure is around, and now if you are stuck somewhere with a flat tire, you will have help and somebody will come and help. And so the same thing I think is happening with computational tools. So yes, for a while you had to really understand every package and every option because that's what you needed to operate that.
Yes.
But I think going forward, what will happen is that there will be more under the hood, and
Hmm.
there will be more help if you get stuck. But at the same time, the tools have to be designed in a way that you can operate longer and be more autonomous and more independent. with the implementation that is available. And I think as much as I care about interdisciplinary education and as much as I care about, understanding what is happening, I think part of that is also on us to make sure that the tools can actually work in this new world correctly. And so then there will be some kind of trade-off, how much of that can be automated and generalized and made it usable enough that people don't have to worry about these tuning parameters, right? And there's some kind of automated kind of selection of options versus what are the options which are really specific to the biological question that you ask. And so I think it is actually very exciting going forward how we can change the design of the tools, also because software development is easier now, right? And
Yes.
so before, so as an academic, a PI, I was quite reluctant to invest a lot of resources into the actual software engineering because PhD students, they don't, get a PhD for software implementation.
Right.
But now it is easier and it's faster. And so we can start worrying about this kind of higher order uh, questions.
Yes.
And I think that overall, the problems for us become more methodological and more kind of general. But at the same time, I'm hoping that our users will be able to benefit from that and kind of do things more rigorously than in the past. So something that is very impactful that happens between identification, quantification, and statistical analysis is data preparation, things like filtering. Let
Yeah.
me remove noisy features.
Mhm.
Let me impute missing values, right? Let me remove something that, I don't believe in, like, oh, I don't know, shared peptides or something like that, right? And so these things are extremely impactful for the downstream conclusions. And this is something that you still do more or less by hand using your own kind of subjective criteria,
Yes.
right? And so one example of kind of creating this automated pipeline is to have more automated ways of saying, what does it mean to have a good filtering, right?
Mm-hmm.
And the different filtering will be appropriate for different types of problems and different types of data. If you have a single cell experiment, and like lots of missing values, it's different than if you have like a bulk experiment, right, with good enrichment. So you would kind of want to filter things differently. So this is one example, right?
Well, you had this great paper last year that I'd wanted to talk to you about. So I feel like you're heading in that direction, which was
Uh-huh.
the title of the paper was Beyond Protein Lists.
Oh, yeah, 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 yeah.
Hey,
This is going beyond kind of interpreting data from a particular technology type, and this is really going in the direction of systems biology. Indeed, when we try to combine data from multiple sources,
yes.
and by data, I don't only mean like measurements, but also evidence from past literature.
Mhm.
When somebody says, well, this protein regulates this protein, well, that's evidence, right? And if it's relevant to our study, well, that's important to integrate. There are many hurdles. And something that also kind of to the date people do more or less by hand is to say, oh, here I have a UniProt ID, here I have some other ID, and here's a gene ID. How do I map all of those things? And look, there's like not one to one. I need to resolve that.
Right,
And
because
then,
one gene gives rise to potentially many proteins,
of
different
course,
splice variants, all the PTMs, et cetera.
of course. And then on the other hand, one paper says this protein regulates that protein, and then the next paper says, no, 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 this protein regulates that protein. And guess what? They may be both right because it's in a different context. And
Yes.
so you need to somehow harmonize all of this information. And so this is another opportunity for essentially treating the language of protein IDs, protein function, or gene protein or biomolecular regulation, treat
Mhm.
it as just another type of language. And so just like we can reason on human language, maybe we can also reason on a language that describes what happens with biological molecules. And there has been quite a bit of progress in that. And I think that this is also a direction that will be very useful. And once we have that, then we can start drawing those causal graphs that I mentioned earlier, saying, well, yes, we have two studies. This is what the studies have in common. This is what the studies have different. This is how we can adjust for the differences. And this
Yeah.
will allow us to make kind of conclusions at a larger level.
Yeah, so these causal graphs, I'd like to go back to an old paper that you and I worked on with Robert, because
That's right.
I think that was a really exciting area of research of really trying to elaborate what are the relationships,
Exactly.
what influences who. And then the part that I thought that you and Robert did that was so beautiful was saying, hey, we have uncertainty about particular relationships. And how can we design the most efficient experiment so we
Resolve
can
that particular area of
yeah,
uncertainty.
exactly,
Yeah.
resolve the particularly uncertain parts of this as much as
Mhm,
possible.
mhm, mhm. That's
And
right. And so, it's funny you mentioned that because so just for the context, Robert Ness was one of my uh, first PhD students in the lab, and it was during the time when I was visiting Prague for my sabbatical, and so it was really wonderful to work together on that. So Robert pretty much seeded an entire direction in the lab, which now leads to that. So how do we interpret the outcomes of proteomic experiments and also multimodal experiments, proteomic, transcriptomic, in a way that goes beyond a list of p-values. I like to say when, we do work that does some hypothesis testing, differential abundance, we have some lists of p-values, and then we give it to our collaborators, and then for 6 months we hear nothing. And this is because it is so hard to move from the p-values to some actual meaningful biology. And I always felt limited because, well, I'm not a biologist. I don't know enough biology to help, but I always wanted to help. And so this is why I'm now very excited by these new developments in parsing natural language as scientific abstracts, parsing ontologies, parsing knowledge bases and data repositories, and creating knowledge bases that are structured and harmonized because now we can compute on them.
mhm.
And if we can compute on them, then we can really take the next step forward and ask questions that are not just mechanistic, is it up or down, but we can ask more like synergetic questions. How do these uh, proteins regulate each other? How do they do it in a particular context? What is similar and what's different in terms of these patterns between data sets, as opposed to just individual proteins changing?
Well, and I think that that point really is that transition from single protein analysis, is this up or down? How confident am I that this is up or down? Really to that more systems perspective of that I
Mhm.
have a chain of multiple events, some of which are short range, short time relationships, and some of which are long distance, long time, potentially across the whole body kinds of relationships.
That's right. That's
And
right.
biology and what it means uh, you know, unfortunately, statistical significance and biological
And
significance
practical significance, yeah.
and
It's not
practical
the,
significance.
yeah. It's not the same at all. That's right. And that kind of goes back to matching the method to the question. Because that's exactly what we just said, right? Because not every question is a differential abundance question. And so then we can ask ourselves, so what is practically significant? What kind of finding actually moves the needle? And then we need to design the methods that answer that question as opposed to somehow trying to fit, the new question to the old framework. And I think that's what's exciting for us is to essentially say, well, we really start from the question. And then the next thing we do, and we do this a lot in the lab, is ask our experimental or like biology or clinical colleagues, okay, how would you do it by hand?
Mhm.
And I remember back at the ISB, that was the question asked a lot. Okay, how do you analyze this data set by hand? And this is very insightful because maybe our collaborators don't need like know the statistical methods, but they certainly have common sense. And
Yes.
they certainly understand the data and they certainly understand kind of the context of their problems that they're trying to solve. And so oftentimes we kind of work backwards a little bit and say, what kind of analysis would give
You're
rise to this type of,
unpacking their intuition and
yeah,
converting their intuition into a statistical framework.
exactly, exactly. And oftentimes the intuition is very meaningful, but it is meaningful in some kind of perfected version of the data, such as, no missing values, no, like perfect distributions, like where averages make sense or where, some kind of simple data summaries make sense. And so the next thing we would do is to say, okay, how would this generalize to a kind of more comprehensive statistical framework, which would account for these edge cases, right? Like for missing values or some kind of, rare types of samples, right? Or rare events or some kind of interferences, so that the intuition that is implemented kind of manually, it becomes a special case of that.
And I think what you're describing is oftentimes when people hear that phrase, okay, what do you do manually? Let
Mhm.
me automate it. What immediately comes to mind is this pipelining and
Exactly.
chaining of tools. But that's
Yeah.
not what you're talking
So
about.
that's very different. That's very different from what we're talking about. We are really trying to create mental abstractions and mental models of that would result in the same analysis if everything goes well. The problem is, of course, in real life, things don't always go as we would like. And so how these mental models can be generalized and extended so that they can actually be practical. And then eventually, yeah, so how do we implement them in a way that people can actually, we don't need to hold everybody's hand to use them so that people can use them independently through large language models, maybe these days.
Yeah. Well, so I think that's a very exciting future. I'm going to put you on the spot and just ask you to dream a little bit and
Okay.
think about your favorite version of the future and the how statistics, machine learning, AI tools are being used in proteomics and biology. And
Mhm.
just share your dream.
Right. Well, I am very intrigued by the artificial cells, right? The models, I don't know what's the word for this, right? So the AI models of how cells work.
Those virtual cells, but the AI generated version, as opposed to
Exactly.
the physics-based virtual cells.
Well, so there are different levels of granularity, right, that you can have, and that's kind of what is interesting. So I'm very intrigued by that because I do think that we can, I kind of in the ideal world, build some kind of expert knowledge system that would mimic, how a cell works. But you're asking me to dream like far away,
I'm
right? So
asking you
we're
to
not,
dream. Yeah,
we're,
I'm
we're,
asking you to
we're we're
dream.
not there. Yeah, yeah, we're not there today, right? And so, and this would include things such as predicting outcomes of perturbations, right? So how the cell works, not just like, in a regular, like steady state manner, but also saying, okay, what if there's a small molecule, right? Or what if there's an external exposure to something, right? Or what if there is a stress? How would the cell respond? I think this will be really incredible in terms of being able to both diagnose the disease and treat the disease and do prevention and just understanding how kind of life works
Yeah.
altogether. Now, we're very far away from that
Yes.
for multiple reasons. One of these reasons is the nature of the data. I know that many of these initiatives that kind of focus on virtual model of the cell, that's what it is.
Mhm.
They focus on virtual models of the cell. They rely on transcriptomic data
Mhm.
because it's cheap, it's structured, right? It's available in large amounts. And as you know, anybody from the proteomics community will tell you, right? So messenger RNA is great, the transcripts are great, but that's not at all the whole story, right? And so I think part of getting to that point is making sure that the data that we have are compatible with, the type of ambition that we have on the AI side of things.
Yeah, if
But
you
at
if
the
you
same,
don't if you don't collect the data that is at the sort of critical linchpin point of a process, then your ability to describe and predict and model that process is poor. You
of
you have to
course.
you have to collect the right data.
Well,
Not
your conclusions are only as good as your evidence, right? And so if we don't have the right evidence at the right, you know, level of resolution, by that I mean the type of molecules, but also temporal resolution and also like spatial subcellular resolution, it is very hard to have the models at the same type of resolution.
Mhm.
And so that's kind of the second part I'm also very excited because even if we had this data, the models that we have currently, and a lot of them are some kind of, neural network based,
Mhm.
models, they're also not enough because even if they can make predictions, they really lack interpretability, explainability.
Yes.
And so it is very difficult to adjust them and adapt them to some new insights. And so I'm actually really excited in the direction of causal AI,
Mhm.
which would incorporate the models of the world, our understanding of the physics and chemistry, and also our understanding of causality into this type of models. And I think this is why, in part, for the moment, uh, these AI models have not been that particularly successful in kind of biomolecular applications as compared to natural language and computer vision,
Yeah,
and
you
other.
it's very clear. There's decades of work in how to take structured textual input and and get data out of it. It's not really clear in biology what is the encoding.
Exactly,
Yeah.
exactly. And what are all the players, right? What are all the, like, let's say we wanted to create some kind of graph that represents the cell. Well, what are the nodes in the graph, right? What is a protein, right? And right, what is it? What is a gene? And like all the other kind of, and then okay, how does it evolve in time? And how do the interactions change in time? So the complexity of this is a whole order of magnitude different from, what people are working with in natural language processing and and even in physical world. And so I'm really excited about this development. Also, part of the challenge is so the data challenge, right? The complexity of AI, and again, the communication. I keep coming back to that because science
Yeah.
is fundamentally done by humans, right? And so it has the limitations that are related to the limitations of humans. And so the complexity of this is hard for us to grasp. And
Mhm.
so, for example, AI folks who want to contribute to this field, they quickly find themselves like, uh, with a very steep learning curve because there is a lot to learn and there's the vocabulary and there is like the type of knowledge that is,
Yeah.
takes time to acquire. And so I think that uh, dreaming about the future, I think part of my dream involves humans interacting in a kind of more mutually complementary manner
Mhm.
and understanding enough of the language of the other field to have the conversation. So maybe not being expert in everything, but understanding enough to listen and understand the nature of the problems and being able to translate these problems into like what they know and into their own area of expertise.
I think that's a really beautiful vision for the future. I think the concept that we have ecosystems of scientists who are highly interdisciplinary ecosystems and are able to complement each other, and the concept that we've gotten to a point where we have the richness and depth and completeness of data to support these really interpretable models that are highly predictive, but not just predictive, but are
Exactly.
understandable. Um, that that would completely be a transformation in the way that we do biology research today. So Olga, for this vision of the future, it depends a lot on having a mix of data and tools and algorithms, et cetera. What is the gap from where we are today?
So I think there's several gaps. So the first is just the scale of the data. Uh, Obviously, these models will have to be very complex. Will have many parameters and many kind of layers of granularity to them, and they need to be supported just with a large extent of evidence. And for the moment, many of these technologies that do not scale to this level. So yes, we can talk about data integration, combining multiple data sets from different investigations, which by itself requires some developments. But even with that, I think it will not be enough because in addition, we need novel types of experiments which would quantify or just characterize the molecules at different level, of at like proteins at single amino acid variation resolution, right? Or different types of post-translational modifications, but also different temporal resolution because the molecules, they're not static, they are dynamic. And so being able to measure them at the right intervals of time and long enough and at the right kind of details of characterization, all of this will require new developments.
Yeah, no, that makes sense. The level of detail that we have available and the scale that we have available are currently insufficient for what we're going to require.
That's right. Absolutely.
So, well, this has been so much fun. Thank you so much for sitting down with me today, Olga.
Thank you for this conversation. It's always fun to talk, dream up like where things can go. And I'm really excited to have a chance to have this conversation. I hope there will be many more.
Absolutely, absolutely. And everyone listening, thank you for joining us on Translating Proteomics, and look forward to seeing you again soon. Don't forget to like and subscribe.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio.